For over a century, the medical consensus surrounding Alzheimer's assumed that when memory loss begins, the brain cells holding those memories are already permanently dead. This belief dictated modern medicine. Billions of dollars and decades of research were channeled into an industrial-scale effort to merely slow the inevitable decay. Patients have operated under the premise that the disease is a one-way street, an unstoppable erasure of the self. But recent studies upend that entire framework. What if the neurons holding your memories aren't dead, but are simply dormant, unplugged, or out of fuel? To test this, researchers first needed to map exactly how a memory is physically built, starting with a simple sensory trigger, like a scent. The smell of fresh wood shavings can instantly conjure a vivid image of your grandfather's workshop. That immediate link between a scent and a past event is called associative memory. We've identified exactly where this connection happens, a brain region called the lateral entorhinal cortex, or LEC. Within the LEC, specialized fan cells fire together. They oscillate in perfect synchrony at 20 to 40 Hz, locking that memory into your internal map. This physical, electrical structure depends entirely on that precise synchrony to persist. In 2021, researchers discovered the trigger for that rhythm. In order for fan cells to encode a new memory, they require a sudden, targeted influx of dopamine. This discovery was unexpected. Dopamine is widely known as the chemical that drives physical motor function and reward pathways, rather than memory formation. Typically, a lack of dopamine results in the physical tremors associated with Parkinson's disease. Seeing it play a critical role in memory was a massive shift in our understanding. Then, in early 2026, a new breakthrough identified a specific failure. Toxic amyloid buildup in early Alzheimer's specifically targets and disrupts the dopamine-producing neurons that feed into the LEC. The memory-holding fan cells don't die right away. Instead, they lose their signal and fall silent. At the same time the dopamine signal is blocked, a second breakdown occurs. There is a severe drop in a critical cellular energy molecule known as NAD+. As the disease progresses, this shortage completely drains the energy reserves of the cells. The neurons remain physically present in the brain, but they are effectively starving, stripped of the power necessary to retrieve a memory. In these early stages, memory loss stems from blocked signals and energy failure, well before the onset of permanent cell death. This presents a compelling hypothesis. If the memory loss is driven by missing dopamine and depleted NAD+, then replacing those elements should flip the memory network back on. Researchers put this to the test using animal models. They administered Levodopa, a medication safely used for Parkinson's for 50 years, to mice with Alzheimer's, successfully restoring their dopamine supply. The results were clear. The mice regained their ability to form and retrieve associative memories, navigating complex environments with high precision. In a separate test, researchers used a specific pharmacological agent to restore the NAD+ energy balance in brains suffering from advanced Alzheimer's. Following the treatment, the brain pathology in these animal models actually repaired itself, leading to a recovery of cognitive function. By restoring the brain's chemical and energy balance, researchers demonstrated that animal models can regain function that was previously thought to be lost forever. These results have so far only been proven in animal models. Moving from the lab to the clinic requires rigorous human trials to ensure these findings translate to patients. But the search is already moving forward. Because Levodopa is a proven, safe medication, clinical trials testing its effect on cognitive slowing in older adults are underway right now. The century-old dogma that death is the starting point of memory loss has been challenged. The signals that translate a scent into a memory may not be gone. They might just be dormant, waiting for the specific physiological conditions required.